Hi, my name is Jonas A Nilsson, and I will present a short video summary of our recent Nature Communications paper entitled Molecular Profiling of Drier Events in Metastatic Uveal Melanoma. And this study has been performed by a multidisciplinary team consisting of surgeons, oncologists, bioinformaticians, immunologists, and tumor biologists like myself. And it has really been spearheaded by a talented bioinformatician called Karlsson, who used to be a PhD student in Karlsson's group and now is a postdoc in my group. So to give you a brief background about uveal melanoma, so uveal melanoma is a rare form of melanoma that arises in the uvea of the eye, most commonly in the choroid or the ciliary body, but in about 1% of cases, it can also present in the iris. So the primary tumor is treated with localized irradiation, or less commonly by removal of the eye. And these treatments can give a good local control, but unfortunately in 50% of patients, metastatic disease will eventually develop, and then mainly to the liver. And once metastasis has been presented, then this disease lacks approved treatments. Different types of immunotherapies, such as adoptive T-cell transfer or bispecific agents, have shown some promising results, whereas immune checkpoint inhibitors to date have not shown much response rates. So targeted therapy with MEK inhibitors did not show any improved overall survival, and BRAF inhibitors, which are commonly used in cutaneous melanoma, cannot be used in uveal melanoma since these tumors do not have BRAF mutations. Rather, uveal melanoma have mutations in GNAC gene 11, or less commonly in CSLTR2 PLCB4, and these four driver oncogenes are mutated in a mutually exclusive manner, which confirms that they are indeed in the same pathway. So prognosis of uveal melanoma is tightly linked to genetics. In fact, the prognosis can be predicted by a gene signature test, which is commercially available. And this test distinguishes between the class 1 tumors, which have a good prognosis, and the class 2 tumors, which have a poor prognosis. Another thing that is associated with high risk of metastasis and poor prognosis is mutations in the tumor suppressor gene BAP1. So most of genetic studies published so far has focused on the primary tumors, including the TCGA studies. And less is known about the genetics of metastatic uveal melanoma. This is partly due to that biopsies of metastasis of uveal melanoma are not always taken in the clinic. But at our hospital, we are using a method called isolated hepatic perfusion. And there's even a trial which is called the Scandium trial, which is a randomized multicenter study that evaluates if this isolated hepatic perfusion increases overall survival in comparison to best alternative care in patients with isolated liver metastasis from uveal melanoma. And during the surgical procedure of isolated hepatic perfusion, the surgeons can take biopsies of the metastasis and give them to the lab for further analysis. And what we've done in this study has been to study the genetics of metastatic uveal melanoma. So we started by just looking at mutations, and what we could observe was that BAP1 mutations exist in over 90% of the metastasis. We also made a novel observation that mutations actually can be present even in introns, creating intronic non-splice site mutations that affect splicing. And we were able to detect these mutations because we had performed whole genome sequencing as well as RNA-seq. We also studied the mutational signatures of metastatic uveal melanoma. And we could observe that most of these uveal melanomas have these signatures that are suggestive of age effects, as well as problems with homologous recombination. But there was one tumor that had an outlier high mutational load and also a different mutational signature. This mutational signature was more similar to cutaneous melanoma, and it was actually the signature 7, which is associated with UV damage. And this was the only iris tumor in the cohort. And in fact, there's no iris tumors at all in the TCGA consortium, which probably is a reason why iris tumors being UV damaged has been missed before. We did obtain a second iris tumor from St. Eric Hospital, and also that tumor had a signature 7 mutational signature, suggesting that the reason why iris melanomas have this UV signature is because they are very superficial in the eye. When we had investigated the mutations, we noticed that one tumor from which we had been able to generate a cell line from a patient-derived xenograft model actually had a BAP1 mutation. So we used retroviruses and transduced either these cells with an empty vector or a vector expressing functional BAP1. By doing this, we were then able to address what were the functional consequence of reintroduction of BAP1. And just as a reminder that the class 1 and 2 are the major transcriptional subtypes in uveal melanoma, and the vast majority of metastases are class 2. And interestingly, when we reintroduced wild-type BAP1 into this deficient patient-derived cell line, these upregulated genes that are normally low in class 2 and downregulated genes that are high in class 2, almost like it switched this tumor to becoming a class 1 tumor. So this was a novel finding suggesting that much of the gene signature associated with these different subclasses could be due to BAP1. We were also interested in looking at the copy number alterations of the metastasis. So what was interesting was that when we compared the copy number changes in the metastasis to that of the TCGA, we could notice that some of these copy number changes were overrepresented in metastasis. And we also obtained 2 primary tumors from which we had also sequenced the metastasis, and we could notice that the loss of 6q, 17p, and gain of 5p were absent in the matched primaries, suggesting that these chromosomal changes are late events or possibly selected for during the metastatic process. Since we had both copy number changes from whole genome sequencing data as well as gene expression from RNA sequencing, we were able to then investigate consistently perturbed genes to see which copy number alterations resulted in gene expression changes, and these then nominate genes that could be interesting for functional follow-up. And when we did this here to the left, you can see this circular plot, which have some of the genes which had either amplifications or deletions, and that this also resulted in changes in transcription. And we functionally followed up some of the genes that had been amplified, resulting in elevated expression of their respective RNAs. And we could see that knockdown of, for instance, PTK2, RAC1, and MAPK14 resulted in a loss of fitness of these cells, suggesting that these could be putative driver genes on amplified chromosomal segments. Finally, we were also interested in looking a little bit about at the immune landscape. So from the metastasis, besides preparing DNA and RNA, we were also able to grow out tumor-infiltrating lymphocytes. So we were interested in then analyzing these to look at cell type representations, clonality, T-cell activation states. And what you can see here in this image on the top row, you can see that the cell types displayed pretty heterogeneous characteristics over different patients. So some patients had a lot of CD4s, whereas others had more CD8s. NK cells were primarily only present in one of the samples. And we could also see that T cells express a wide range of immune checkpoint receptors. And besides PD-1 and CTLA-4, also expression of TIM3, TIGIT, and LAG3 was prominent in these TILs. And finally, we wanted to address whether or not these tumor-infiltrating lymphocytes could be functional. So we used tetramer assay, which is a soluble form of HLA to which you can bind different types of peptides. And in this assay, we addressed whether or not T cells in these cultures could react with tetramers binding either MART1 or GP100 to well-known melanoma-associated antigens. And indeed, we were able to identify antigen-specific tumor-infiltrating lymphocytes. And this can actually be an explanation why certain patients also respond to adoptive T-cell transfer in the clinic. So with this, I would just like to end and thank all the co-authors for their strong work, and also the patients and their families for generously participating in the Scandium trial and in this study, and the physicians and nurses involved in recruitment and treatment of patients in the Scandium trial, the Genome Medicine Center in Gothenburg and SciLife Labs for sequencing, and technical support from Karina and Sofia, as well as funding from various funding sources. And I also want to thank you for tuning in. Thank you.